Today's video we are going to talk about carbohydrates. Carbohydrates are the hydrated carbons. Carbohydrates. And they are made up of carbon, hydrogen and oxygen. Okay, and we can classify carbohydrates into three types as monosaccharide, disaccharide and polysaccharide. So monosaccharides are the basic unit of a carbohydrate. Now let's see some details on monosaccharides. So monosaccharides, depending on the number of carbon atoms, the name of the monosaccharide varies. If the number of carbon is 3, we call it triose. If it is 5, it's pentose. And if it is 6, it's hexose. Okay, so triose, they are helpful in respiration. You both learn about this in detail in your A2. And pentose, they are part of DNA. You know, the deoxyribose sugar. And in RNA, ribose sugar. These two are pentoses, as the number of carbon there is 5. And hexose, there are several examples you should know. One is glucose, is the most common one, and the fructose and galactose. Okay, now we are going to talk glucose. You know, glucose, there are 2 different types of glucose you should know about. And first one is alpha glucose. Okay, now let's see how an alpha glucose looks like. The glucose are in ring shapes in nature. Okay, so this is the hexose ring. They have an oxygen in the corner and 1, 2, 3, 4, 5 carbons, and the 6th carbon is up here. Okay, now let's see how we can draw the structure. Right opposite to the 5th carbon you have 3rd carbon. In the 3rd carbon we have OH at the top, and in all the other carbons OH are at the bottom. Okay, and now I can complete the structure by filling the hydrogens in there. Okay, so in the 1st carbon, if you look at the structure again, the OH is at the bottom and the H is at the top. Now let's see how a beta glucose looks like. So it's very similar to the alpha glucose, only the difference is these H and OH swaps. OH goes to the top and the H goes to the bottom. So I can draw the structure. Can draw the numbers 1, 2, 3, 4, 5 and the 6th carbon here. And in the 3rd carbon again, the OH at the top and all the others OH at the bottom. But now in the 1st carbon, OH is going to be at the top. I can complete the structure. by filling hydrogen here. So yeah, in the first carbon now the OH is at the top, and we compare with the alpha glucose. Yeah, that's simple. That's the basic difference with alpha glucose and beta glucose. Yeah. And now we are going to move to disaccharides. Yeah, disaccharides is made up of two monosaccharides. So two monosaccharides bind with glycosidic bond by condensation reaction. Now let's see how they form. Okay. So yeah. Okay, so these are two alpha glucose molecules. If you look at the structure here, the OH is at the bottom, and in here the fourth carbon. This is in the first carbon, here is fourth carbon. So in the condensation reaction we should lose one water molecule. So OH group from one carbon, the first carbon, and H group from the third carbon is released, one water molecule is released, and then they form the 1,4 glycosidic bond, which form the disaccharide. In here, as these two molecules are glucose molecules, the disaccharide formed is maltose. If there are two glucose molecules bind together with 1,4 glycosidic bond, the disaccharide formed is maltose. Okay, and there are several other disaccharides. You should know about the monosaccharides of those. Yeah, so as I said now, if there are two glucose molecules form glycosidic bond between them and they produce the maltose, and if one glucose and one fructose bind together, they will form sucrose. This is another disaccharide. And if one glucose and one galactose bind together, they will form lactose. Lactose, that's in the milk sugar. Yeah, that is disaccharides. Now let's see some polysaccharides. Polysaccharides is the polymer of monosaccharides. So you should know about several polysaccharides. First, you should know about amylose, amylopectin, starch, glycogen, and cellulose. These are the polysaccharides you should know about. Okay, now let's see one by one. First, let's talk about amylose. Yeah, amylose, the basic unit of amylose is alpha glucose, and they join together by 1,4 glycosidic bond. Okay, so for example, yeah. polysaccharides, the chain extends. So there are no branch, as it has only one type of glycosidic bond. The polysaccharide is in long chain. Okay. Yeah. Now the second example is amylopectin. Okay, so in amylopectin, again the basic unit or the monomer, the monosaccharide is alpha glucose, but they have two types of bonds, one for glycosidic and they have 1,6-glycosidic, which leads to the branching pattern of amylopectin. So let's say if the chain is lengthened by 1,4-glycosidic bond, like this, another molecule can bind here to the first carbon and the sixth carbon of a chain. So this is the sixth carbon. Okay, so in some places the chain will bind to the sixth carbon, which is the 1,6-glycosidic bond. So now the structure look like the chains with branch. Okay, so yeah, so the amylopectin has branches. Okay, that is amylopectin. Okay, now we are moving to starch. So yeah, starch, there's no a single compound called starch. The starch is made up of both the amylose and amylopectin. Yeah, so if we take a starch molecule, 30% of starch is made up of amylose and amylopectin 70%. So now you can tell the characteristic of starch molecule as we know about amylose and amylopectin. So if we talk about the bond types, you know, in amylose they have 1,4-glycosidic bond, and amylopectin they have 1,4, and also in addition to that they also have 1,6-glycosidic bond. Okay, now let's see what's the function of starch in living organisms. You know, starch is the storage molecule in plants. So what characteristics of starch leads to this function or leads to the storage ability? You know, starch is insoluble, so that's the basic characteristic which lead to the storage function, because if it is insoluble they don't affect the osmotic potential of the cell, and also the bonds in the molecule have stored the energy, and it is easy for them to hydrolyze and produce and release the energy when they need it. So that is starch. Now we are moving to the fourth polysaccharide, glycogen. You know, glycogen is the storage molecule in animals. Okay, before going into the functionality, we discuss about the structure. Yeah, glycogen is very similar to amylopectin. So if I say similar to amylopectin, I think now you can list the characteristics of amylopectin. What's the bond type they have? They have 1,4 and 1,6 glycosidic bonds. So as they have 1,6 glycosidic bond, they are branch structure. So they have branches. Okay, and now let's see what's the character, or what are the characteristics of glycogen as a storage molecule. Now, they are the storage molecule in animals. So yeah, the characteristics are similar to that of starch. So they are insoluble. So the effect to osmotic potential is very less or not. So yeah. And another thing I should say here is, I said it is similar to the structure of amylopectin. So how we can differentiate this amylopectin from glycogen is, there are two things. One is the glycogen is very long compared to amylopectin, and they have more branches than amylopectin. So yeah, that's the basic difference between glycogen and amylopectin. Okay, so the final polysaccharide is cellulose. Okay, keep in mind, up to now we talk about amylose, amylopectin, starch and glycogen. These four molecules, or these four polysaccharides, are made up of alpha glucose. Okay, but in here the cellulose, the basic unit or the monosaccharide is beta glucose. The only molecule you have beta glucose as monosaccharide is cellulose. Okay, yeah. And in beta glucose they have the bond type 1,4 glycosidic bond. They have only 1,4 glycosidic bond. So again we can say this is a long chain. There's no branches. Okay, so you know the cellulose is, or acts as a structural component of plants in the cell wall. They provide the rigidity for the cells. So how they provide this rigidity? Yeah, cellulose, you know, the monomer is beta glucose. Okay, they form the bonds between two molecules. If you look at here, I have drawn only a rough diagram. The two monomers, when they bind together by a 1,4 glycosidic bond, they are in inverse conditions, like they are not in straight. If one is the oxygen molecule face upwards, in the other molecule oxygen face downwards. Okay, this adds to their rigidity as well. And another thing we should discuss is the long chain of cellulose molecules. They bind with adjacent cellulose molecules by hydrogen bonds. Okay, and then these cellulose molecules form a fibril. Okay, and then these fibril, microfibrils, and lots of microfibrils bind together to form macrofibrils, which leads to its rigidity and the strength. Okay. Yeah, now let's see an overview of what we have done on the polysaccharides.